In this final part, part 4 of our EMG module, we're going to start by conducting a very brief review of the knee extensor muscle group, because it's this muscle group that will be used as an example of how the EMG signal can be used with motion and force data to get a better understanding of human biomechanics. So the quadriceps femoris muscle group we know is made up of 4 muscles. It has an origin on the anterior inferior iliac spine as well as the femur, and inserts onto the tibial tubercle via the quadriceps tendon and the patellar tendon. It's innervated by the femoral nerve, and we're going to focus our examination on the vastus medialis, vastus lateralis, and rectus femoris muscles, because these are the 3 superficial portions of the quadriceps femoris muscle group. The vastus intermedius, of course, is positioned deep to the rectus femoris. And so this particular study sought to examine the pattern of muscle activation as a function of force level. And so participants in this study, as you can see here in this photo, this is just a sample photo from the internet, but this is a device that was used to sample isometric knee extensor force production following or during 3 maximal voluntary contractions. So participants, 30 participants in this particular study, performed 3 maximal 5-second contractions, very brief efforts, to establish their MVC. They then performed a series of 9 submaximal knee extensor contractions in a random order that ranged between 10 and 90% of that maximum voluntary contractions. And so they performed light contractions, near maximal contractions. These were 5-second contractions, all conducted at a 60-degree knee angle, so not like the photo here. The knee was actually flexed a little bit more. And they were able to achieve those submaximal forces through concurrent visual feedback from a computer monitor, so they only produced enough force to match their force line to a target on the screen. And so the figure on the right is what is often referred to as the EMG force relationship. Along the x-axis, we have the force produced by the knee extensor muscle group, but notice that all the forces produced by the participants, because the strength capabilities varied among all 30 of the healthy young adults, all the relative levels of force all the absolute force levels were converted to a percent of the maximal voluntary contraction, which was taken as 100. Likewise, the sampled EMG data were quantified and normalized to the EMG bursts that were quantified during the 100% MVC that would then yield a unit in that percent unit. And so what we see here are recordings from the 3 superficial knee extensor muscles, which act as synergists, so they all produce force to carry out the same function, and that's to generate knee extension. And so the clear illustration here is that as force increases, the recorded EMGs which occur during the production of those forces increases in a relatively proportional manner. There's some nonlinearity here, but for the most part, this is a rather proportional increase in the EMG. And notably, at near maximal levels, at 90%, what we see is a convergence of the activation of all 3 superficial muscles to almost equal levels, whereas there was some differences throughout the submaximal range. In terms of using the EMG to quantify muscle fatigue, what we see here is a very simple way to examine muscle fatigue, is to have a person perform an isometric contraction. So we're going to sample knee extensor torque at some prescribed level, sub-maximal level of their maximal voluntary contraction. And so this person would be performing a sub-maximal isometric contraction for as long as they possibly could until they reached what's called a failure point, a point at which they could no longer sustain a prescribed knee extensor force level. And so what we saw in a study such as this, by simultaneously recording the electrical activity, the EMGs from those muscles, that'll give us an indication of whatever pattern might exist in the EMG when the force level is held relatively constant. There's going to be a little bit of variability around the target, but for the most part, this is a constant isometric force level, and we're going to examine what happens to the EMG signal. So in this particular study, 17 healthy young adults performed that isometric knee extension at a very high level of intensity, 80% of their maximal voluntary contraction. The average contraction duration was approximately 43 seconds, and the overall finding was that there was an increase in the activation, in the EMG signal throughout the entirety of the contraction time. In fact, the activation of the vastus lateralis was observed to be greater than the vastus medialis during the entirety of the contraction. I'll touch on the perceived exertion response shortly, but essentially here, the perceived exertion was the subjective rating on a 10-point scale reported by the research participants throughout the contraction. And so the graph on the right illustrates the increase in EMG. So the contraction time was normalized so that we could compare all the research participants, where 100% represented the failure point. And what we see here is the normalized EMG, the magnitude of the EMG normalized to the maximal voluntary contraction. So at the very beginning, we would expect to see EMG values somewhat around the force level at 80% MVC force level, and so we do see that the EMG, the activation was around that level. But as they sustained the contraction, it's important to remember the force level was the same, but the EMG increased and then hit this plateau level towards the end of the contraction, suggesting to us that as that muscle starts to fatigue, the nervous system will increase the activation, will increase the drive to the muscle that's reflected in the electrical activity in order to compensate for muscle fibers that may be dropping out and not able to generate the necessary force to hold that target. On the right-hand side, we see that there was a progressive increase in the reported perceived exertion of the research participants that followed a fairly similar pattern. There was a rapid increase followed by a relative plateau, suggesting to us that there is a link between the activation patterns in our muscles and how people subjectively perceive those types of contractions. This is another study that sought to examine, instead of isometric contractions, dynamic contractions. So 30 participants, 15 men, 15 women, were recruited into the study, and instead of performing that maximal isometric voluntary contraction, the participants were evaluated for something called a 1 repetition maximum for a single leg knee extension task. And so in this setup, we see that the 3 superficial knee extensor muscles were evaluated and the EMGs were sampled. What we see, this contraption here is an electrogoniometer that was used to track changes in knee position. And following the establishment of how much weight a person could lift while only being able to perform 1 repetition, they then performed 1 set of repeated knee extension contractions at a load that was 50% of their 1 RM, to the point where they couldn't move the weight anymore. And so overall, the repetitions to failure was fairly similar. Women were able to perform on average, one more repetition than the men at 50% MVC. And so this tracing is a sample tracing of only one knee extensor contraction. And so we see the electrogoniometer changing as the person moves through the knee extension phase. That's the concentric phase of the state of the muscle. The participants were required to hold that knee extended position for 2 to 3 seconds and then to slowly lower that weight back to the starting position and then continue to repeat that until they couldn't perform the task anymore. And so what we see here is that in terms of the kinematics, we have knee extension, holding knee extension in a static position, and then a knee flexion movement, but that was controlled by the knee extensor muscle group. The concentric, isometric, eccentric state refers to the actual length of the muscle and the change in length. So the first observation to make here is that we see a relatively large burst of EMG activity from the 3 superficial quadriceps femoris muscles as the knee goes through knee extension, held fairly consistent throughout the isometric phase, and we see relatively lower amplitudes as the weight is slowly lowered back to the start position because the need to activate the muscle decreases because the force output is augmented by the lengthening state of the muscle. And so this figure here illustrates the change in EMG in the activation patterns of the muscles throughout the entirety of that one set of 50% 1 RM repetitions to failure. And this graph only illustrates the concentric phase. So along the x-axis, this is the contraction duration as a percent because all the participants performed a different set of repetitions. Those numbers had to be converted to a percent. The normalized EMG was converted to a value that is a percent of the 1 repetition maximum. And as we can see here, during the concentric phase, in a very similar manner that we observed with the isometric type of contraction in the previous research study, there was a nonlinear increase in the EMG of all muscles. And so there was a rapid increase in all 3 superficial muscles, followed by this plateau. The greatest increase that seemed to continue to plateau the least was the largest of the 4, which is the strongest of the 4, which is the vastus lateralis muscle. So the question here is, despite the fact that the same load was moved with each repetition, why did the amplitude, the time domain EMG increase? Fatigue mechanisms were obviously at play that caused the central nervous system to increase its drive to the muscle that resulted in an increased activation as some muscle fibers started to essentially drop out and unable to perform the same level of force. So this reflects an increasing neural drive to the muscle. This figure here from the same study illustrates what happened during the isometric phase. And so during the isometric phase, across contraction duration, we see very little change from repetition to repetition. In fact, one muscle seemed to have, the vastus medialis here, seemed to have drifted downwards in terms of its activation slightly. So overall, EMG, the increase was the overall amplitude, the normalized amplitude was greatest for the VL, which is greater than the VM, which is greater than the rectus femoris. And so the question here is, why was there no change during the entirety of the contraction? This is somewhat speculative. But technically the isometric phase might be an easier part of the exercise. And in a mechanical sense, it's probably not that much easier than the concentric phase because of the fact that the moment arm of the load was greatest during the isometric phase, but what we see to hold that position repetition to repetition, the muscle was fairly consistent. Now, this figure from the same study examines the EMG signal in the frequency domain. So if you recall from one of the other parts of this module, one way to examine the EMG signal is to convert the time domain signal into the frequency domain signal, and then to use statistics to characterize that positively skewed distribution. So the median frequency was sampled from each repetition and expressed as a function of contraction duration. And so what we see here is a very, very typical response in the frequency profile of the EMG signal, that as fatigue ensues, there's a progressive and fairly linear decrease in the median frequency. That suggests to us that the frequency starts to drift towards lower values. So the rate of the decrease was seen to be greatest for the rectus femoris, less so for the VL, and less so for the vastus medialis. And so, as we saw with the concentric phase, fatigue mechanisms will explain this drift towards a lower frequency. One of the major factors that will cause this drift towards the lower frequency is a decrease in what's called the conduction velocity. That's how fast the action potential spreads across the muscle fiber membrane. And so as that muscle fiber starts to fatigue, the ability of the muscle fiber to restore ion distribution will start to decrease, and that'll prolong the action potential length, which is inversely proportional to its frequency, and that's often seen as a major factor that contributes to this downward shift in the EMG frequency. The EMG can also be used to evaluate various reflexes. So we'll start this review of the spinal reflex. So the spinal reflex is an inherent built-in mechanism in the neuromuscular system that's designed to detect and regulate changes in muscle length, as well as the rate of change in muscle length. And so this is a stepwise process that as whole muscle lengthens, that whole muscle will stretch intrafusal and extrafusal muscle fibers. The extrafusal muscle fiber is the muscle fiber we talk about all the time. Those are the force-generating muscle fibers. The intrafusal muscle fibers is contained within something called the muscle spindle, which is a length sensor in the muscle. So the muscle spindle is also lengthened when the muscle lengthens. The equatorial region, which is the middle part of the muscle spindle, is stretched, and that generates what are called 1A afferent impulses. Action potentials are generated within sensory neurons built inside the muscle spindle that are fit under this category called 1A sensory neurons, and they send action potentials very quickly to the spinal cord, informing the central nervous system that the muscle is lengthening. Those sensory neurons will synapse with alpha motor neurons, which are the lower motor neurons we discussed in the first part of this module, and that will result in a direct activation of the motor unit. And so what we see here is an illustration of a reflex hammer hitting a muscle tendon. And so even though the magnitude of the stretch is very small, it is very rapid. So that tells us that the muscle spindles are particularly sensitive to how fast a muscle stretches. And so the muscle spindle contains sensory neurons. The most prominent one here is what we mentioned just a minute ago, the 1A sensory neuron. The 1A afferent will send action potentials up towards the spinal cord and will synapse directly onto alpha motor neurons of the same muscle. and that's referred to as the homonymous muscle. So the homonymous muscle is the muscle that receives the motor input from which the original sensory input was generated. And so this is a tracing of an EMG signal during a reflex test. And so on the very bottom, we see what's called the tap. This was generated by a force hammer. And so this tells us that the force hammer hit the patellar tendon. This tracing is the same electrogoniometer tracing. So after the patellar tendon was impacted by the hammer, we see the knee start to move. It starts to go through knee extension, knee flexion, knee extension in the manner we see on this cartoon illustration. And so this was sampled from the same synergists with the mechanical stimulus, and of course we see what's called an oscillating decay of knee position or knee range of motion, where it results in this extension followed by a flexion, and the leg will continue to swing until it no longer swings, much like a pendulum. But what we see with that reflexive activation, the leg will only move. It will only move when the superficial, let's go back here. When the superficial knee extensor muscles are activated, and you see these very short duration, high level, high amplitude bursts from the superficial knee extensor muscles. But it's very difficult to examine the relationship among all of these tracings until we zoom in. And so the illustration here is a little bit grainy, but I think we'll be able to see what is exactly happening in terms of the tracing and how we can match it to the physiological events. And so what we see here with this green line is when the hammer made initial contact with the patellar tendon, and we have a very, very short period of time at which the muscle, in this case it's the rectus femoris, generates an electrical burst. That's called the latency period. And that's the time between the stimulus, which is the hammer hitting the tendon, and the measured electrical response. So this period of time, what we have is the muscle is being stretched, the sensory neurons are activated, we have a reflex in the spinal cord, results in motor end plate action potentials, and eventually muscle fiber action potentials. And notice that the EMG signal will decay and come back to baseline levels, and there isn't even any movement of the leg into knee extension. So the time between the initial activation of the muscle and the mechanical response is called the electromechanical delay. And that is approximately 25 milliseconds. That period of time does vary by muscle and by person, but 25 milliseconds is a reasonable estimate for the electromechanical delay. And the electromechanical delay incorporates action potential, muscle fiber conduction, calcium release from the sarcoplasmic reticulum, and cross-bridge cycling. So when we examine the EMG signal, the force produced by the muscle, in this case, we're not measuring force, we're measuring motion, will occur at a slightly later point in time. They're not superimposed on each other. And the total time is what's called the reflex time. That's the latency plus the electromechanical delay. So this ends the fourth part of our EMG module.